This section describes the separation of plant pigments using a technique called paper chromatography. This technique relies on the fact that all photosynthetic organisms contain pigments. Pigments are receptors of the light energy that is needed to drive photosynthesis. Photosynthesis, as you have learned, is a process by which plants use light energy to convert inorganic carbon in the form of carbon dioxide to organic carbon in the form of sugars. There are four main pigments that we are concerned with for this experiment. Chlorophyll A is found in all photosynthetic organisms. This pigment may be the most familiar to you, as it imparts the dark green color in most of the leaves you see. Chlorophyll A, as you have learned, has two specific absorption maxima, or light wavelengths, that are preferentially absorbed. These wavelengths are in the blue-violet region and the red region. The chlorophyll A pigment does not absorb and therefore reflects light in the green wavelengths, which is why many plants look green to the eye. However, there are three other important pigments contained in most plants. They are chlorophyll B, xanthophylls, and carotenoids. These are also referred to as accessory pigments. Chlorophyll B is an accessory pigment because it can absorb light energy from different wavelengths than chlorophyll A, and then transfer this energy to chlorophyll A. Xanthophylls and carotenoids are accessory pigments for a different reason. These pigments often act as sunscreens for the plant. They absorb excess energy in the blue and green, or high-energy light wavelengths, and serve to protect the plant from excess light. Just like humans, plants can get a sunburn of sorts and be harmed by UV light produced by the sun. Sometimes these plants will look red if left in very high light. This is typical of plants that produce excess carotenoids, which are red in color during times of light stress. Each of these pigments is a different molecular weight or size, which allows them to travel at different rates through a piece of chromatography paper. In this way, we can separate them to determine the pigment composition of a plant. Now that you have reviewed the principles of pigment and paper chromatography, it is time to assemble the materials that will be used in this experiment. Remember, the purpose of this technique is to determine the types of pigments found in common plants. For this experiment, we will be using prepared extracts of leaves of spinach or coleus. You will also need the following to perform this experiment: a solvent like petroleum ether, acetone, a chromatography chamber with a few millimeters of solvent in the bottom, chromatography paper cut to fit the chamber. Be careful to only touch the sides of the paper. This is so that you do not get grease or dirt on the front of the paper, as it will affect your results. And glass capillary tubes to put very precise drops of pigment extract onto the paper. Once you have assembled these materials, you will need to draw a line using a pencil a few millimeters from the bottom of your paper. This line will help you measure the rate of flow of your solvent and pigment. Once you have drawn this line, carefully make a pigment drop using the capillary tube. You must apply a very small amount, let it dry, and then repeat often until you have a dark stain of pigment. This will assure good results. And remember, make sure your pigment drop stays well above the pencil line. When this is complete and your pigments have dried, carefully place the paper inside the chamber, making sure that your solvent depth in the chamber is below the pencil line. If it is too deep, remove some with a pipette until it is a suitable level. Replace the lid on the chamber and allow the solvent to move up the paper until it's within a few millimeters of the top. This will take about 10 to 15 minutes. While we are waiting for the chamber to develop, it would be a good time to discuss how to calculate your results from this exercise. The results of this exercise will be examined in two ways. The first is referred to as the RF value, or rate of flow. The equation used to calculate the rate of flow is RF equals distance of pigment migration divided by distance of solvent migration. First, draw a line at the center of your solvent front. Then, circle each pigment and mark the center. These are easily measured using a metric ruler and then plugged into the equation for each pigment. Also, make a note of the color and relative location of each pigment and try to identify which pigment group they belong to. Carotenoids at the top of the paper, orange. Xanthophylls in the middle of the paper may be in two bands, yellow. Chlorophyll A below xanthophylls, bright blue-green. Chlorophyll B below chlorophyll A, khaki green. Anthocyanins will not move from original location, reddish purple. Remember, each plant may contain different pigments. Now that we have discussed how to interpret your results, take your chromatography paper out of the chamber and mark the pigments and solvent front and take the appropriate measurements. Let's discuss the pigment bands you have on your chromatography paper. This is an example from the coleus extract. Notice how the anthocyanins did not move from the original pigment spot. This extract contains from the bottom up 5 pigments, anthocyanins at the bottom, then chlorophyll B, which is yellow-green, then chlorophyll A, which is blue-green, then the yellow xanthophylls, and finally the carotenoids, which are orange. Now let's look at the spinach results. This extract is missing the anthocyanins, but from the bottom to top has chlorophyll B, chlorophyll A, xanthophylls, and a faint band of carotenoids.